In today's lecture, we're going to discuss the key functions and features and structures associated with cellular membranes. So we're going to focus mostly on the plasma membrane, the membrane that surrounds the cells. Plasma membrane is said to have a fluid mosaic character. What does that mean? It means that in the fluid mosaic model, the components of the membrane, such as phospholipids, cholesterol, proteins, or carbohydrates, can move laterally within the membrane. That mosaic doesn't mean that the membrane can be dissolved in the extracellular or intracellular fluid. All it says is that the components of the membrane are not tethered to a particular site and can move around the membrane. In other words, the membrane has the fluid mosaic structure. Four main components, four biological macromolecules that comprise the principal components of the plasma membrane are phospholipids, cholesterol, proteins, and carbohydrates. Membrane is roughly 5 to 10 nanometers in thickness, and the main components that form more than 50% of the plasma membrane are lipids. Lipids that comprise plasma membrane include phospholipids and cholesterol. Second component of the membrane by percentage, proteins. Many membrane components carry modifications. Lipids and proteins are often modified by the addition of carbohydrates, which results in the formation of glycolipids and glycoproteins. Now, it is useful to recall what phospholipid consists of. A typical phospholipid molecule consists of glycerol, two fatty acid residues connected to the glycerol moiety, and the phosphate head group that is also connected to the glycerol. Cholesterol is located in the plasma membrane alongside with the phospholipids, and the main function of cholesterol, you can see cholesterol molecules here, here, and here. The main function of cholesterol molecules is to regulate the rigidity of the plasma membrane. On this illustration, you can clearly see phospholipid bilayer with a whole bunch of individual phospholipids forming this bilayer together. You can clearly see proteins, integral, as well as peripheral. You can also see that some lipids are modified by the addition of carbohydrates forming glycolipids, and many proteins also carry carbohydrate residues which are essential for these proteins' structure. Now, a typical phospholipid consists of a glycerol backbone that is connected to fatty acids and the phosphate group. Here you can see the two fatty acid residues, one saturated and one unsaturated, are connected to two of three glycerol carbons. Phosphate group with the substituent R which can be of different sorts. For instance, ethanol amine is attached to the third carbon of glycerol. Now the head, the part of the phospholipid molecule highlighted here in pink, is polar, and it has a negative charge. The tail, on the other hand, tail that is formed by two fatty acid residues, is nonpolar. Tails in the membrane, as you can see here, form the hydrophobic core of the membrane. You can see that majority of the membrane is hydrophobic, while the surfaces are hydrophilic and charged. Now, if you would arrange phospholipids so that the hydrophobic parts would face each other, they can form a structure known as mycel. In some cases, you can actually produce a structure called a liposome on the left, which looks like a spherical phospholipid bilayer. So the important feature of this phospholipid bilayer arrangement is that hydrophilic parts that form membrane surface effectively interact with the water-based, water-containing outside and inside of the membrane. In other words, hydrophilic parts of the membrane establish energetically preferable interactions with water in the extracellular fluid or in the environment, and with water molecules inside of the cell cytoplasm. Now, second major component of the plasma membrane are proteins. Proteins can be integral or peripheral. Integral proteins span through the plasma membrane. Some examples of integral proteins are shown in this picture. You can see that it can be one fragment of a protein spanning through the membrane. It can be several parts of the protein going back and forth through the membrane, or it may be multiple parts of the protein forming the transmembrane domain. The membrane spanning regions of the proteins is unsurprisingly hydrophobic, because they have to interact with the hydrophobic core of the membrane. You can see on this illustration here transmembrane integral proteins spanning the membrane, and most likely this part of each protein will be composed of hydrophobic amino acids. Proteins are oriented along phospholipids, and as we mentioned before, hydrophobic regions are spanning within the membrane, while hydrophilic regions extend from the membrane and establish a physical contact with the cytosol on the inside of the cell, or with the extracellular fluid on the outside of the cell. Peripheral proteins, unlike integral, are attached to the membrane surfaces. For instance, here you can see a peripheral protein. These proteins can be attached to phospholipids, or in some cases to other integral proteins. The function of peripheral proteins on the inside of the cell is primarily serving as enzymes, providing the attachment of the membrane to the cytoskeleton, or serve as signaling molecules. On the outside of the cell, proteins, along with carbohydrates, form recognition sites. Turns out that carbohydrates, being a third key component of the plasma membranes, form a layer of carbohydrates on the outside called glycocalyx. So these carbohydrate fragments on the surface of the cell represent a part of the glycocalyx. Glycocalyx, composed of carbohydrates attached to either proteins or lipids, is essential for the cell recognition. The specific set of glycoproteins or glycolipids on the surface of the cell allows cells of the immune system to distinguish between self and non-self cells. So, how does a membrane regulate its own fluidity? And broadly speaking, what are the factors that regulate the fluidity of the membrane? Well, first of all, the membrane itself, as we mentioned before, is not rigid. However, it maintains its structure pretty successfully. The membrane is fluid. And this is the key statement about the properties of the plasma membrane, is that it is rigid in the sense that it's not dissociating as the cell grows, but at the same time, the components of the membrane can flow laterally within the membrane. If you penetrate plasma membrane with a needle and then retract the needle back, the membrane will, quote-unquote, heal itself. It will seal behind the needle, and no special repair activities are required for this. Considering the fact that lipids and proteins within the membrane are separate and not rigidly attached to each other, it is conceivable that these molecules can move and float within the membrane. Now, what makes the membrane more or less rigid? Turns out it greatly depends on the composition of the phospholipid tails. Phospholipid tails that contain saturated fatty acids, shown here, these tails are pretty densely packed and make the membrane more viscous. In other words, the presence of saturated fatty acids in the membrane explains how this membrane can be more rigid and less fluid. On the other hand, membranes that contain greater proportion of unsaturated fatty acids are generally more fluid. If you look at this, you can see that kinked chains due to the presence of the cis double bond in unsaturated fatty acid. That increases fluidity of the membrane. So there's a bit of a typo here. So what happens to the membrane when temperature increases or decreases? So it turns out when a temperature decreases, saturated fatty acids, because of their compression, will result in the membrane that is dense and fairly rigid. On the other hand, if the same temperature is reduced in the membrane with more unsaturated fatty acids. The kinks in the membrane will maintain the membrane fluidity. Different organisms contain different proportion of the saturated and unsaturated fatty acids, which explains the ability of the membranes in those organisms to adjust to the temperature changes. In other words, microorganisms that exist in environments with higher temperature will contain phospholipids with more saturated fatty acids. So, if an organism normally exists in a greater temperature, it will contain greater proportion of saturated fatty acids. Now, organisms that have to be exposed to the lower temperature will contain greater proportion of unsaturated fatty acids, because as the temperature drops, these kinks in the membrane, kinks in the fatty acid tails, these kinks allow cells to maintain the fluidity of the membrane despite of the reduced temperature. Now, plasma membranes, one of the key properties of the plasma membranes is that they are selectively permeable. What does that mean? It means that not everything can get in or out of the cell. Another important feature of plasma membranes is that they are asymmetric. If you would look at the outer surface of the membrane in this illustration, you can see that there are a lot of carbohydrates on the outer surface of the membrane, but not on the inner one. There will be some proteins or carbohydrates that are present only on the outer surface, and just as like, there will be some proteins that are present on the inner surface, but not on the outer surface. Moreover, peripheral proteins that are found on the inside of the membrane have different functions compared to the proteins found on the outside of the membrane. As we mentioned before, carbohydrates are found on the exterior but not on the interior surface. All of this explains the phenomenon of asymmetricity of a plasma membrane. One of the key properties of plasma membrane is that it is amphiphilic. Amphi means both, philic means love. What it means is that plasma membrane has hydrophilic, these, and hydrophobic, these regions. This complex organization with hydrophilic regions of the membrane facing extracellular milieu, extracellular fluid, and intracellular cytosol, and the hydrophobic part of the membrane located between two thin hydrophilic layers, explains the feature of the selective permeability of the plasma membrane. Turns out that nonpolar, small, lipid-soluble molecules can cross the membrane directly. Examples of such molecules are respiratory gases, relatively small molecules of fat-soluble vitamins, and various fat-soluble drugs. Substances that are polar or hydrophilic can't pass across the membrane directly. They cannot squeeze between individual molecules of phospholipids and instead have to use transmembrane proteins, channels, carriers, or pumps. Examples of such polar hydrophilic substances include various ions, which are always hydrophilic because of the charge, amino acids, proteins, peptides, carbohydrates, and nucleotides. So now it's time to talk about the phenomenon of diffusion. Diffusion, the whole process of diffusion, is a passive process. So what does that mean, passive? Passive simply means that the substance that diffuses moves down its concentration gradient. You can see at the bottom I have an illustration of this. The substance moves down its concentration gradient from high concentration to low concentration. Now, the definition of a gradient is the uneven distribution of concentrations across the space. If you look at this illustration, you can see that the concentration of a hypothetical chemical in the extracellular fluid is much greater than the concentration of the same chemical in the cytoplasm. The difference in concentrations represents a gradient, and this gradient drives the movement of the molecules across the membrane. Diffusion stops when the concentrations of the chemical are equal on both sides of the membrane, equal across a space. In other words, diffusion stops when the gradient of concentrations doesn't exist anymore. The term passive refers to the fact that no energy is required for diffusion, since the chemical moves from its high concentration to its low concentration, kind of like a bowling ball can move down the slope. However, the term passive doesn't necessarily imply that this process of diffusion cannot be regulated and occurs uncontrollably across the membrane. Before we move on with our discussion of what types of diffusion we can observe in the cells, let's consider the factors that increase the diffusion rate across the membrane. Steeper gradient, first and foremost, will increase the rate at which molecules move across the membrane, which makes sense if you look at this illustration. The steeper slope will increase the rate of diffusion. Smaller size of the molecules. Molecules that are smaller will move across the membrane faster, well, because they are smaller. Increased temperature, which increases the velocity of individual molecules, will also increase the rate of diffusion. Lower density of the solvent that surrounds the membrane, and lower polarity, and it should be more hydrophobic. Lower polarity or greater hydrophobicity will increase the diffusion rate. Membranes with higher surface area will demonstrate greater rate of diffusion, and membranes that are thinner will also show a greater rate of diffusion. Now, factors that decrease diffusion rate across the membrane is the greater distance that has to be traveled by the molecules when they cross the membrane. Now let's see how polar or hydrophilic molecules can cross the membrane. Such molecules can cross the membrane using so-called facilitated diffusion or facilitated transport. There are a couple of ways the molecules can cross, the hydrophilic molecules can cross the membrane using facilitated diffusion. They can either use channels or they can use carriers. So in case of facilitated diffusion, molecules cross the membrane along the concentration gradient via transmembrane proteins. The problem with the transport of polar molecules is that they're going to be repelled by the hydrophobic core of the membrane. This is why they can't cross the membrane directly. So molecules that need to cross the membrane bind to the receptors on the surface of the protein here, and they pass through the integral proteins that we call channels. Channels are specific for the substance that they transport. In other words, you can see two different substances on the exterior side of the cell, but only the green hexagon can be passed through that particular channel. In other words, sodium channel will allow the passage of sodium, but not potassium or calcium, for example, and vice versa. So channel on the inside is hydrophilic. It's important to note because we just discussed that these parts of the protein, the parts of the protein that interact with hydrophobic core of the membrane, are essentially hydrophobic. However, this part of the channel, the inside of the passage, is hydrophilic, and it provides the pathway which is hydrated essentially, pathway that contains plenty of water molecules. So channels can be open all the time. However, the majority of the channels are gated. Gating of channels means that they can open and close in response to chemical signals such as neurotransmitters, voltage changes, depolarization or repolarization of the membrane, altering the membrane potential. They can open and close in response to mechanical stimuli and so on. The second type of transmembrane proteins that can facilitate the transport of substances across the membrane are carrier proteins. Carrier proteins bind the substance, you can see it right here, and then alter their own shape, which allows them to move the molecule across the membrane. Carriers are usually specific for a particular substance, and just like channels, they allow the passage of the substances across the membrane in either direction. So what determines the direction of the passage? It is a gradient. If the gradient looks like this for that green circle, green oval, then this molecule will be diffusing from the extracellular fluid into the cytoplasm. However, if we would flip the concentrations and increase the concentration of green oval on the cytoplasmic side, so that it would become higher than the concentration of the oval on the extracellular side, the direction of the diffusion will be reversed. The rate of the diffusion that's mediated by carriers is limited. First and foremost, it is limited by the number of carrier molecules in the membrane. Since there are only so many carriers, it doesn't matter how steep the gradient is. The rate of the diffusion will remain constant because increased steepness of the gradient doesn't matter. You only have so many carriers. Another important feature of carriers is that they are dramatically slower compared to the channels. We're talking about 10 times slower. Now that we have figured out the mechanisms for the passive transport for the diffusion, it's time to talk about the phenomenon of osmosis. So osmosis is the movement of water through a semi-permeable membrane. In other words, water moves through the membrane that is permeable for water, but not for the solutes. Essentially, in this case, water diffuses along its concentration gradient, which is inversely proportional to the concentration of the solutes. In other words, the water moves from the area with the low solute concentration to the area with the high solute concentration. Now, it's important to understand that osmosis depends on the concentration of all the substances, but not on the nature of the substances. So, when you look at the left beaker, the volume of the solution on both sides of the semi-permeable membrane is the same. The concentration of the solutes is different. So it means that the amount of water on the left is greater than the amount of water on the right. Since membrane is permeable only to water, but not to the solutes, water will diffuse down its concentration gradient. So that's the concentration gradient for the water. And it will move to the right half of the beaker. And you can see that as water diffuses towards a higher concentration of solutes, the level of the fluid on the right side of the membrane increases. So osmosis will continue either until the water gradient disappears, just like in any diffusion, or if the hydrostatic pressure of the solution on the right will equilibrate the diffusion of water from left to right. In other words, when hydrostatic pressure of the solution on the right equals the osmotic pressure generated by the flow of water, we can represent it using arrows. So arrow from left to right represents osmotic pressure, while the arrow from right to left represents hydrostatic pressure. Now, with the concept of osmosis comes the concept of tonicity. Tonicity is determined by the osmolarity of the solution. Osmolarity is the total solute concentration. It's important to understand that osmolarity does not depend on the chemical composition of the solutes. So it can be sodium chloride or potassium chloride. Now, when a solution has a low osmolarity, it means that there is a lot of water relatively to the number of solute particles. If osmolarity of the solution increases, then the relative ratio of water molecules to the solute particles will decrease. In other words, solutions with high osmolarity have greater overall concentration of all the solutes. Water moves across the semi-permeable membrane from the side of the membrane with lower osmolarity to the side with the higher osmolarity, which allows us to define the concept of hypertonic, hypotonic, and isotonic solutions. Hypertonic solution is the one where the concentration of solutes, in other words, where the osmolarity outside is higher than osmolarity inside. Now, isotonic solution is the one where osmolarity outside equals the osmolarity inside. And hypotonic solution is the one in which osmolarity outside is less than osmolarity inside. Now, if you look at the ratio of osmolarities in the hypertonic solution, stating that outside osmolarity is greater than the inside, water will move out of the cell. In case of hypotonic solution, water will move into the cell. And in case of isotonic solution, there will be no net movement of water. Now, if you think about this, in hypertonic solution, water leaving the cell will result in cell shrinkage. We call this crenation or plasmolysis. In isotonic solution, the cell will remain unchanged. But what happens to the cell in a hypotonic solution? You can see that cell swells. Now, if cell will continue swelling, it may lead to cell lysis. However, in organisms like plants, fungi, and bacteria, cell wall prevents a cell from lysis. All those organisms, fungi, plants, and bacteria, are always slightly hypertonic to the cellular environment. In other words, they exist in hypotonic solution. That constant small influx of water into the cell allows the plant, fungal, and bacterial cells to maintain turgor pressure. This turgor pressure is so important that it is one of the key structural mechanisms to support non-woody plants, such as grass, for example, upright. Now, a lot of protozoa exist in the environment that is hypotonic, which means water moves into the cells. So how can protozoa that do not have cell walls survive in such hypotonic environment? The answer is pretty simple. They pump the water out of the cell constantly. They use enormous amount of energy for this, but nevertheless, they can survive by constantly pumping the water using contractile vacuoles. Now, bacteria can survive in the environments with high concentrations of salt, which by all means should be hypertonic, but apparently they aren't. So hyper and hypotonic are two relative terms, which means bacteria that survive in the environments with high concentration of salt, in other words, environments with high osmolarity, maintain high osmolarity inside the cell. So the osmolarities of cytoplasm and of the extracellular environment are the same. This knowledge about osmolarity and about the ability of hypertonic environment to essentially suck the water out of the cells, resulting in the plasmolysis or crenation, is important for the food industry because we use salt as the preservation chemical, since salt prevents multiple microorganisms from growing, from reproducing. It essentially kills them. So far we've only discussed the movement of chemicals along the gradient. So what about the movement of chemicals against the gradient? This can be achieved by the means of the active transport. Active transport requires energy. In the most cases, in the form of ATP. In active transport, substances are usually moved against the gradient. So here you can see the example of sodium-potassium pump, which moves potassium that is located from the left. It moves potassium from the left side to the right side, and it moves sodium on the right side to the left side. But first we need to figure out what determines the charge on the membrane, because membranes are charged. Turns out that the proteins that stay in the cell, and proteins are negatively charged, these letters A represent proteins, that creates a net negative charge on the intracellular part of the membrane and positive charge on the extracellular part of the membrane. In other words, the interior of the cell is electrically negative in respect to the extracellular fluid. That creates the gradient of charges across the membrane. Now we know it has been measured that the intracellular concentrations of potassium are greater than the extracellular ones, as you can see on the illustration below. On the other hand, concentrations of sodium are greater in the extracellular fluid. So overall, the diffusion of sodium will be driven into the cell by both the negative charges and the concentration gradient. So sodium diffusion into the cell is driven by both concentration and electrical gradients into the cell. On the other hand, the electrical gradient drives potassium into the cell. Potassium is positively charged, and the intracellular space is negatively charged. But its concentration gradient drives it out of the cell. So all the combination of all the concentration and electrical gradients form what we call an electrochemical gradient across the membrane. Which ions have greater concentration outside of the cell? That would be sodium, calcium, bicarbonate ion, and chloride anion. Inside of the cell, the concentrations of potassium, magnesium, hydrophosphate, and sulfate are greater than their concentrations outside of the cell. So in active transport, as we mentioned before, ions are moved across the membrane against their concentration gradient. And we already mentioned that active transport depends on the ATP. A good example of active transport is a sodium-potassium pump or sodium-potassium ATPase. This enzyme moves potassium ions into the cell and moves sodium ions out of the cell in the ratio of 2 to 3. In other words, per every 2 ions of potassium moved into the cell, 3 ions of sodium are moved out of the cell. This is a very important protein in human cells since it maintains the electrochemical gradient across the membrane and establishes and maintains proper concentrations of potassium and sodium on both sides of the membrane. So it all starts with 3 sodium ions binding to the interior part of the protein. You can see it right here. Hydrolysis, breakdown of the ATP, releases energy that is used to deliver those 3 sodium ions outside of the cell. Now, inorganic phosphate remains attached to the pump, which results in the change of the pump shape and reorients itself towards the exterior of the cell. When this shape change occurs, the affinity, the ability to bind sodium ions for this pump decreases, so sodium ions can now leave the pump. So here you can see the illustration of sodium ions being transported against the concentration gradient with the use of ATP. Now, the affinity of the pump to potassium ions, on the other hand, in this particular conformation increases. So potassium ions bind to the pump, phosphate detaches from the pump. That detachment of the phosphate results in the shape change. Pump reorients itself towards the interior, and the affinity for potassium ions goes down. Two potassium ions move into the cytoplasm, and we go back to the square one. So, as a summary, for every 3 sodiums that are moved out, 2 potassiums are moved in. The net loss of positive charges from the interior of the cell, every 3 positive charges move out and only 2 positive charges move in. That makes this pump electrogenic. It allows cell to maintain its electrical gradient. Now, the phenomenon of secondary active transport is very common, particularly in the human kidney. So in this case, the active transport here uses the energy of diffusion that occurs down the concentration gradient to move ions or any other compounds against their concentration gradient. So we know that sodium ion concentration outside of the cell is higher than inside. This is because of the primary active transport, sodium-potassium pump, that we just discussed. It means that overall electrochemical gradient moves sodium ions right here into the cell. These ions, of course, are then pumped back out into the outside of the cell. So this is not energy is not generated out of nowhere. Energy is actually spent on the maintenance of high sodium concentrations outside of the cell. But anyway, diffusion of sodium into the cell provides energy for the transport of other substances, for example, glucose or amino acids, into the cell. Another possible use for the energy of diffusion is the use of high energy ions in the mitochondria to generate ATP. The potential energy of the gradient of hydrogen ions is converted into the kinetic energy of the diffusion when they cross the membrane using ATP synthase. ATP synthase utilizes that energy to synthesize ATP from ADP and inorganic phosphate. Another example of secondary active transport is here. Proton pump maintains high concentration of hydrogen ions outside of the cell. Diffusion of hydrogen into the cell through the so-called sucrose hydrogen cotransporter helps to move sucrose from its lower concentration outside of the cell towards its greater concentration inside of the cell. Now, when sodium or hydrogen move in the same direction as the molecule that they facilitate the transport for, we call this co-transporter or sym-porter. But when the directions are opposite, we call these molecules anti-porters. What if we need to bring something bigger into the cell? Something like a bacterium or a fragment of another cell. For this, we can use a set of techniques known together as endocytosis. Endocytosis is simply a movement of large molecule cellular parts and other whole cells into a cell. First type of endocytosis that we're going to discuss is phagocytosis. Phagocytosis depends on the protein collatrin, which stabilizes the section of the membrane about to form so-called pseudopodia. These pseudopodia surround the particle, for instance bacterial cell, forming a pocket. This pocket pinches off, forming phagosome, which eventually merges with a lysosome, forming so-called phagolysosome, which is highly acidic and contains hydrolytic enzymes and acid that are capable of breaking down whatever this particle is. Another mechanism for what we call a bulk transport is pinocytosis, or cell drinking. In this case, cellular membrane invaginates and collects whatever is in the extracellular fluid. This is why we call it drinking. This process is not really specific. The vesicle that is formed as the result of pinocytosis is much smaller than a phagosome and does not merge with a lysosome at all. A subtype of pinocytosis is podocytosis, when the protein caveolin stabilizes the fragment of the membrane, and that results in the formation of a stable vesicle called caveolae. These vesicles are usually smaller than the ones formed in pinocytosis. Podocytosis allows cells to not only transport molecules or ions or whatever is outside of the cell into the cell, but also allows molecules to cross the cell and emerge on the other side, so-called transcytosis. Podocytosis is pretty common in the formation of vesicles that transport different molecules between membrane-bound organelles within the cell. The last type of bulk transport that we're going to talk about is receptor-mediated endocytosis. This process is extremely important in a specific uptake of various molecules and sometimes even microorganisms by the cell. Here you can see receptors on the extracellular surface of the membrane that interact specifically with molecules or virus particles outside of the cell. When such interaction happens, the protein called clathrin stabilizes the vesicle and helps to select cargo for that vesicle. So such stabilized clathrin-coated vesicle can take up whatever these stars represent and bring it into the cell. Many human pathogens, such as bacterial toxins and viruses, use this process to enter the cell. Now, exocytosis is the type of the bulk transport that doesn't bring the compounds in, but rather moves the compounds out. It can be crucial for the release of various waste into the extracellular space. Neurons release neurotransmitters by the mechanism of exocytosis, where a vesicle fuses to the membrane, plasma membrane of the cell and releases its content in the extracellular space. This table summarizes different types of transport: diffusion, osmosis, facilitated diffusion, primary and secondary active transport, and 3 types of the bulk transport. Please know whether the transport is passive or active, in other words, whether it requires energy or not, and what kind of material can be transported in these processes. Now we're going to shift gears and move into the concept of metabolism and bioenergetics. So metabolism is the sum of all chemical reactions inside of the cell. These reactions can include breakdown of complex molecules or their synthesis. Some of these reactions are spontaneous and release energy. Other reactions require energy to proceed, therefore cells must constantly obtain energy. The major energy source for many cells is glucose, because it stores an enormous amount of chemical energy within its bonds. These bonds are broken down during the process of cellular respiration, releasing energy that can be converted into ATP. Another important process that converts one type of energy into another is photosynthesis. In photosynthesis, the light energy is converted into the energy of chemical bonds in the molecule of glucose, which is essentially synthesized from the atmospheric carbon dioxide. On the diagram on the left, you can clearly see the first reaction, the reaction of photosynthesis, that uses solar energy to produce glucose and oxygen. The organisms that can do it are producers. In other words, the role of producers is to carry out photosynthesis and convert light energy into the energy of chemical bonds. Like these acorns contain a lot of chemical bonds. The energy of chemical bonds is then used in the process of cellular respiration by consumers, such as animals, and decomposers, which destroy dead organisms. So, when energy of chemical bonds is released during cellular respiration, it is used to produce ATP. ATP serves as the energy currency of the cell, and I want to highlight this. ATP is not a molecule that stores energy for the long time. It is not a savings account. It's cash that constantly changes its owners. If an organism needs to store energy of chemical bonds, in other words, if an organism needs to store glucose, organism can convert it to starch if it's a plant, or to glycogen if it's an animal. All metabolism in the cell can be described as the series of metabolic pathways. These metabolic pathways contain a bunch of biochemical reactions that are connected to each other. And essentially the goal of those reactions is to convert some kind of a substrate molecule into a product. Pathways that build complex molecules, like the one on the bottom, are called anabolic. Pathways that break down complex molecules are called catabolic. Metabolism essentially consists of anabolic and catabolic pathways. For the most part, anabolic pathways require energy and therefore are called endergonic. Energy is required to synthesize a complex molecule from the smaller subunits. Catabolic processes, on the other hand, usually release energy and therefore are called exergonic. When complex molecules are degraded into their building blocks, these processes usually release energy. So if one wants to store energy, it can be stored by producing complex molecules, since this process requires energy input. When these complex molecules, such as carbohydrates or fats, are used to produce ATP, when they are catabolized, that catabolism releases energy which can be used to produce ATP. It's important to note that metabolic reactions in the cells are not spontaneous. In other words, they can't occur without catalysis by the enzymes. Enzymes in the cell facilitate or catalyze all reactions, anabolic or catabolic, exergonic or endergonic. Now, before we talk about
some more about metabolism and enzymes, we need to define what is energy. So energy is the ability to do work. Energy can be of a different type. It can be electrical energy, light energy, heat energy. But perhaps the most important division between two types of energy is potential and kinetic. So potential energy, illustrated by this water behind the dam, refers to the potential of the object to do work. This water doesn't do anything, it just stays behind the dam. But if this dam will be open, then this water will start moving. That movement, that object in motion, has a kinetic energy. A car, a moving water, a moving flying bullet. All of these objects have kinetic energy. Now, energy is constantly transferred. It can be transferred from potential to kinetic when you open the dam. You can use your force to move the spring, to compress it, so convert kinetic energy of your hands into potential energy of the compressed spring. Chemical energy can be converted into kinetic. For example, chemical energy of chemical bonds is considered to have a potential to do work. Therefore, by and large, molecules contain enormous amount of potential energy. And in this example, molecules of hydrocarbons contain potential energy that can be converted by the engine into the kinetic energy of a moving car. How can we quantitate the energy changes during the chemical reactions? For this, we're using a concept of the Gibbs free energy. Gibbs free energy reflects the energy change when we move from reagents to the reaction products. All energy transfers in the cell involve the loss of some energy in its unusable form, meaning losing its heat, so-called entropy. Gibbs free energy is the energy available after accounting for that heat loss, for that entropy. In other words, Gibbs free energy is the usable energy that is available for doing work. Every chemical reaction has a change in free energy, or delta G, which can be quantitated as an enthalpy of the reaction, change in the enthalpy of the reaction, minus T delta S, where delta S is the change of the entropy within the system during the reaction. T is the temperature. If the energy in the reaction is released, then the change in the free Gibbs energy is negative. That negative sign reflects that the energy during the reaction is released. In other words, the products have less free energy than the reactants, and such reactions are spontaneous. In other words, they proceed without any addition of an energy. If energy is used during the chemical reaction, the change of the free Gibbs energy is positive. This reaction is endergonic. The products of the reaction have more free energy than the reactants, and such reaction is not spontaneous since it requires the input of energy. As we mentioned before, anabolic processes are usually endergonic, and the change of the Gibbs free energy for these processes is usually positive. Catabolic processes are, generally speaking, exergonic, so the change of the Gibbs free energy for such processes is usually negative. Every reaction is characterized by the activation energy. Activation energy is the amount of energy that is required for the reaction to proceed. In other words, activation energy is this hump right here. During the reaction, chemical bonds break and other chemical bonds form, in order for some bonds to break and others to form, the molecule must be brought into so-called transition state. So this transition state right here, in order to bring molecule into transition state, energy is required. And you can see that transition state of the reaction always has a higher energy than the reactants. So unlike the change of the Gibbs free energy, activation energy is always positive. Activation energy is required to increase the frequency and the force of the collisions between the molecules during the reaction. Usually it's a heat energy from the surrounding. Activation energy essentially determines the rate of the chemical reactions. The greater the activation energy, the slower the reaction. Usually, for most cellular reaction, activation energy is too high for them to proceed at the reasonable rate. So in order to reduce the activation energy, in order to increase the rates of cellular reactions, cells use catalysis, more specifically the catalysis by the enzymes. Before we get to the enzymes, let's discuss the main physics laws that regulate the energy transfer, so-called laws of thermodynamics. The total amount of energy in the universe is constant, period. Therefore, according to the first law of thermodynamics, energy can be transferred, it can be transformed, but it can't be created or destroyed. In other words, chemical energy stored in food can be converted into kinetic energy of movement. Chemical energy, this energy transfer from, chemical energy to kinetic energy, just one of the examples. Another example is the conversion of light energy into chemical energy of leaves and other materials in the plants. So another law that we need to be aware of is second law of thermodynamics, which states that in every energy transfer, some amount of energy is lost in the form that is unstable. Usually, energy is lost as heat. So heat energy is defined as the energy that transfers from one system to another, but does not do the work. So during cellular metabolism, energy is lost as heat. This is what allows us, warm-blooded creatures, to maintain our body temperature. Now, the more energy a system loses as a heat to its surrounding, the less ordered and the more random the system. In other words, the chaos in the system increases. So entropy is the measurement of the randomness, measurement of chaos, disorder within the system. Greater entropy refers to greater disorder and lower energy of the system. All energy transfers lose some of their usable energy, which means every energy transfer inevitably increases the entropy in the universe and the disorder in the universe. Now, the key molecule that is responsible for energy transfers in the cell is adenosine triphosphate, or ATP. It's a relatively simple molecule which stores potential energy in its phosphate bonds. ATP is the primary energy currency of the cells. It powers the majority of cellular reactions and makes it easy for cellular reaction to retrieve energy. Imagine that you would have only carbohydrates, lipids, and proteins as energy sources. So each reaction would derive energy from each of these molecules, which means during each reaction you should have a bunch of enzymes, some enzymes that are capable of transferring energy from carbohydrates into the reaction, other enzymes that are capable of transferring energy from lipids into the reaction, and so on and so forth. Use of ATP increases the versatility of energy transfer in the cell. The structure of ATP is relatively simple. The nucleoside adenosine on the right, which contains a nitrogenous-based adenine and the ribose sugar, is connected to 3 phosphate groups named alpha, beta, and gamma, closest to the furthest from the ribose. Each 2 phosphates are linked by high-energy bonds. These 2 bonds have the greatest energy, okay? And when these bonds are broken, they release an enormous amount of energy. So typical products of breaking ATP are ADP, adenosine diphosphate, and inorganic phosphate. These 2 products of the reaction have much lower free Gibbs energy, which means this reaction is exergonic. Now, when ATP has to be synthesized, ADP and phosphate are combined with the input of energy, and in this anabolic and ergonic reaction, ATP is produced. ATP molecule is highly unstable, so if it's not used in the cellular process, it can dissociate spontaneously into adenosine diphosphate and inorganic phosphate, releasing energy that is lost as heat. However, controlled hydrolysis of ATP provides energy to pretty much all cellular processes in what we call energy coupling. You can see it here at the bottom. Solar or chemical energy facilitates the formation of ATP. Energy stored in the phosphate bonds of ATP is used for cellular work. For instance, anabolic reactions. When this energy is used, ATP is hydrolyzed into adenosine diphosphate and phosphate. These 2 components are then recombined into ATP with the input of energy, and the cycle continues over and over. So what are those enzymes that facilitate chemical reactions? Enzymes are catalysts. Most of them are proteins, however, some enzymes are RNA molecules. Enzymes bind reactants and distort them to break the bonds and reform the bonds, and that bond breaking and formation is much easier when enzymes are involved. It's important to remember that enzymes do not change the delta G of the reaction, as you can see in this graph. The purple graph represents the process without the enzyme, without the catalysis, and the red dashed line represents the process with catalysis, with the enzyme. You can see that the only parameter that they change is activation energy. Enzymes reduce the activation energy without altering the ratio or the yield of the products. The enzyme here binds the substrate and converts it into the products. When enzyme binds the substrate, it distorts it and therefore lowers the activation energy of the reaction. In some cases, enzymes can form covalent bonds with the substrate molecules. But enzymes never wasted in the reaction. They remain unchanged. They return to the original state once the reaction is complete. You can clearly see it that the enzyme in the beginning and at the end of the reaction are the same. The substrate binds to the active site of the enzyme, and since each active site has a unique set of amino acids, that explains very high specificity of the enzymes. Active site of an individual enzyme will be specific to its own specific substrate. There is some flexibility to the rule, but there is not much. Every enzyme has optimal conditions for its operation. These optimal conditions include pH, temperature, concentration of ions, and so forth. What's important to remember, enzymes can work outside of the optimal conditions, but their performance will be worse. So how can we regulate the enzyme activities? Turns out that there are various molecules that can stimulate the enzyme function or inhibit it. So inhibitors can be competitive and non-competitive. In case of a competitive inhibitor, it binds to the active site because the inhibitor is similar to the substrate. When an inhibitor is bound to the active site of an enzyme, it blocks the substrate from binding to that active site. During a non-competitive inhibition, also known as allosteric inhibition, the inhibitor binds to an allosteric site, a site that is different from the active site. You can see example here on the left when inhibitor binds to the enzyme and inhibits it. Now, how does an allosteric inhibitor work? When it binds to an allosteric site, it changes the shape of the active site so that a substrate cannot bind to it anymore. Now, allosteric activators bind to the location that is not an active site either, but they stabilize the active form of the enzyme and allow it to perform its function. In other words, allosteric activation are essential for the maintenance of the shape of the active site that is conducive for the affinity of the enzyme to its substrates. Many enzymes require cofactors and coenzymes. Cofactors are ions such as iron, zinc, magnesium, copper, molybdenum, and so on, while coenzymes are usually organic molecules such as vitamins. These vitamins listed here are all coenzymes for different, well, enzymes. When a cofactor binds to the enzyme, we call this inactive form without a cofactor an apoenzyme. However, when apoenzyme binds the cofactor, it forms a complete form of the enzyme called holoenzyme, which is now active. Another way to regulate the activity of the enzyme is through so-called feedback inhibition. In feedback inhibition, the product of the reaction regulates its own production. So more products will slower their production. Look at the scheme at the bottom. In this illustration, the product of the pathway binds to the first enzyme in the pathway and prevents it from binding the substrate. Now, this is another example how isoleucine inhibits its own production. When isoleucine binds to an allosteric site of the enzyme called threonine deaminase stops being active and cannot initiate the process, resulting in the formation of that same isoleucine. ATP serves as an effective allosteric regulator, inhibitor, or activator of many enzymes involved in the metabolism of glucose. Generally speaking, the abundance of ATP results in the reduction of its synthesis. In other words, ATP inhibits many reactions in the process of glucose metabolism, slowing down ATP production. On the other hand, ADP, which is the product of ATP breakdown, if you remember, works as the positive regulator for many enzymes that in turn inhibited by ATP. When ADP levels are high compared to ATP, cell produces more ATP. When ATP levels are higher than the levels of ADP, ATP synthesis is inhibited. Feedback inhibition is one of the key regulatory mechanisms for the enzyme activities.